So yung enzymes naman na yun mag-degrade ng mga smaller peptides into uh, amino acid. Okay? So that's what happens in our stomach before in order to form your amino acid. So this is more example of illness or disease that may happen in our body related to the formation of amino acid is acute pancreatitis. So this disease caused by obstruction of the normal pathway by which pancreatic secretion enter the intestine, causing excruciating pain and damage to the organ that can prove fatal. So what happens here? So since yung may mga acidic may mga enzymes ka that break down your peptides, it can possibly break down also yung inyong uh, walls of cells. And if ever na walang available na inhibitor within the walls of your intestine or stomach, you can feel pancreatitis. Okay? And it can damage your pancreas. So, um, so that's what happens in acute pancreatitis. Okay? Alright. So after it enters the body, magpa-process na yung different processes for your amino acid. Okay, so this is the overview of what happened in our amino acid oxidation. So you have your intracellular protein and dietary protein, which will undergo formation or cleaving to form your amino acid. This amino acid will be cleaved, leaving your amino group and your carbon skeleton. So this is what happens generally in your amino acid. Ang una muna niyang ginagawa is to cleave the amino group. Then your carbon skeleton will undergo to different catalytic processes in order for it to prepare for the different or for the other catabolic pathway. Specifically for citric acid, pwede rin for glucogenate, glycolysis. Okay? So how about yung amino group mo naman will undergo different processes, and most specifically is the formation of carbamyl phosphate in order it to enter for your urea cycle. And urea cycle will be the formation of urea, which is the toxic, or which is the excretion product or the waste product in form of urine. Okay? So this is the overview of the what happens in the amino acid catabolic pathway. Okay? So we will get into details about that. So what happens first is the removal of the amino group of your amino acids. All right. So you have initially here your amino acid ingested in protein. Ayan. And inside the liver, what happens here is the transfer of your amine group to one from another to another um, ketogenic compound, which is alpha ketoglutarate, and then your formation of your carbon skeleton. Right? Yung carbon skeleton na to will be move forward to form pyruvate or any other starting material for different catabolic pathway. And ang pinakamalaga dito is yung removal of the toxic amine group. So this is ammonium ion. So yung ammonia mo, this is a toxic to body and it should be wasted. And it will undergo urea cycle for the excretion of it. It will be either excreted as ammonia itself or urea or uric acid. That depends on the organism na mag-excrete mo. Okay? And, ayan. Okay? So excretory forms of nitrogen, ayan. So this is either in form of ammonia or form of urea or form of uric acid. So si ammonia, mostly it is excreted by aquatic vertebrates. So this happens by meron ata dito sa yeah. So yeah. So this is just the form or the structure of ammonia, urea, and uric acid. So how can they be, or what organism release or excretes this specific form of or amino group? Plants conserve almost all of the nitrogen, as we have mentioned earlier, since they use it for formation of different metabolite used for formation of different metabolites. While many aquatic vertebrates release ammonia to the environment by either passive diffusion from epithelial cell or active transport via gills. Okay? So many terrestrial vertebrates and sharks excrete nitrogen in the form of urea. So for example, of us humans, we excrete urea in form of urine, and urea is far less toxic than ammonia and has a very high solubility in water. Okay? Some animals, such as birds and reptiles, excrete nitrogen as uric acid. Uric acid is rather insoluble. Excretion as paste allows the animal to conserve water. Kaya yung mga birds and reptiles, if you can see their feces, mostly ay parang paste form since ang uric acid is not insoluble, and they are in need to conserve water, kaya ganun yung mga feces nila. Humans and great apes excrete both urea and uric acid from purines. So yung purines, ito ay from your DNA or RNA, mga nucleic acids. Okay? Right. Right. Ito. This is what happens first in amino acid oxidation. So nangyayari is the transamination or transfer of your amino acid or amino group. Yan. Amino group using the pyridoxal phosphate cofactor and the catalyst amino transferring. So what happens here is just you transfer uh, from your amino acid, the amine group, to alpha ketoglutarate, ayan, to form your keto acid. L-glutamine acts as a temporary storage of nitrogen, and L-glutamine can donate the amino group when needed for amino acid biosynthesis. Okay? So, pwede naman siyang gamitin ni glutamate to transfer this amine group na meron siya to form amino acid if ever na kailangan ng body. Okay? So what is pyridoxal phosphate and pyridoxamine phosphate? So these are the cofactors needed for the transamination. These are intermediate and enzyme-bound carrier of amino groups. Aldehyde form can react irreversibly with amino groups, while aminated form can react reversibly with carbonyl group. So yun lang. You can either use both if ever kung ano yung need nung body to form. Right? So pyridoxal phosphate is covalently linked to the enzyme in the resting enzyme by an internal aldemide or shift base. Your shift base is C double bond N, and here. This is usually happens in different nitrogen and carbon reaction, and especially in amino acid formation. The linkage is made via a nucleophilic attack of the amine group of an active site. So si amine group will, ayan, here. The amine group will be act as a nucleophile. Yung extra lone pair niya will be the nucleophile, and it will attack the electrophilic carbon of the uh, of this pyridoxal phosphate, and abstract yung carbon oxygen bond. And now you have your ship base when in removal of your water. Okay, so this is just an example of the enzyme that happened or enzyme involved in the formation of ship bees. Okay, this is the overview of what happens to the pyridoxal phosphate and your amino acid. So it can either go to formation of keto acids or the formation of another amino acid and amino. Okay, so in details, ayan. The external albumin of PLP is a good electron sink, allowing removal of alpha hydrogen. So this just happened. This is just a view on formation of your keto acid using the pyridoxamine phosphate in the transamination phase. Okay. And so the pyridoxamine, formation of pyridoxal phosphate or pyridoxamine phosphate containing na yung nitrogen. What happens here? Yeah. Formation of external albumin with substrate. So remember that this happens within an enzyme. So may bees that can abstract yung hydrogen, alpha hydrogen, and you can form a shift base. Ito na yung shift base mo. Okay? Abstraction of proton leading to quinoid formation. And now, kailangan i-restore nung iyong uh, nitrogen-containing ring, yung kanyang aromatic property. So, the movement of the electron will happen and it will grab a proton to have this um, compound. And upon hydrolysis or removal of or no, not removal, addition of water, now you have your keto acid and your pentoxamine phosphate. Okay? So yung shift base mo can furtherly go to another path, which is here. Ayan, it can grab proton or babalik siya dun sa kanyang happens here. Nag-form lang ulit si pyridoxal phosphate. Okay. PLP also catalyzes racemization of amino acid. Okay. So what happens here is just the change of configuration of your L-amino acid to D-amino acid. Okay. So you just ano, change the configuration of the amino acid. Pyridoxal phosphate also can catalyze the carboxylation of amino acid. Ayan, as we have mentioned earlier, you can either the aminase or the carboxylate your amino acid. Now you have an amino. Okay? So either of the two, it can PLP can help us in three specific pathways. Depende dun sa need ng body and depende dun sa ano yung yun nga, kung ano yung kailangan pathway na susunod. Okay? Removal of amino group by glutamate dehydrogenase. So oxidative deamination occurs within mitochondrial matrix. So what happens naman dun sa glutamate mo na nag-capture ng amino group? So using NADHP, you can form another shift base. And this shift base, upon addition or hydration, you can now release your amino group. Kasi temporary lang na storage si glutamate ng iyong amino group. At yung kinapture niyang amino group from your dietary amino acid. Okay? So, afterwards, itong ammonia na ito will undergo to your urea cycle. Urea cycle. And itong alpha-ketoglutarate will undergo again to different or to another capture of amine group mula sa another batch naman ng amino acid. Okay? So nagre-recycle lang yung alpha-ketoglutarate to glutamate in order para maging efficient yung removal of your amino group. Pathway for ammonia excretion is trans-deamination. So that means lang na transamination and oxidative deamination. Okay? Ammonia is safely transported in the bloodstream as glutamine. Yun din yung mahalaga. Kaya mahalaga na ma-convert yung amino group into glutamine dahil iniiwasan ng body na magkaroon ng free ammonia sa labas ng cells niya or sa labas, yeah, sa bloodstream. Kasi ammonia is a toxic compound. Ammonia is toxic compound. Okay? Excess glutamine is processed in intestine, kidneys, and liver. So you can see here, your glutamate will capture, ayan, ayan, meron ko ng L-glutamine containing amine group, and it can now move out from your to your bloodstream, and it can now be processed at ma-release yung uh, ammonia. Ammonia transfer of glutamate to pyruvate to make alanine. Okay? So this is a process naman between the liver and the muscles. Vigorously working muscles operate nearly anaerobically and rely on glycolysis for energy. And glycolysis yields pyruvate. If not eliminated, lactic acid will build up. And this pyruvate can be converted to alanine for transport into liver. Okay? So kapag masyadong maraming pyruvate, it can be converted to alanine using your glutamate. Ayan. And yeah. And you transfer your amino group to your pyruvate to form alanine, and this alanine can enter the liver para mag-undergo naman ng transamination with alpha-ketoglutarate in the liver, and it will build a cycle. Okay, so depende sa kailangang need ng energy ng iyong muscles, especially kapag you are vigorously working, yung mga nag-workout or yung mga uh, in competition na kailangan ng body. Ayun. So that is the source of your energy for the muscle contraction and movement. Okay? Ito, but this is what happens sa iyong nitrogen once it was removed from your amino acid. Right? Ammonia is recaptured via synthesis of carbamyl phosphate. So, mahalaga na carbamyl phosphate, ay wala pa nakalagay dito. Pero carbamyl phosphate is the allowed form of the amino group, amine containing para makalabas siya dun sa mitochondria. Okay, so ammonia is captured by synthesis of carbamyl phosphate. This is the first nitrogen acquiring reaction of the urea cycle. So before mag-proceed to urea cycle yung compounds mo, it should be converted to carbamyl phosphate. So what happens here is your bicarbonate, excuse me, okay, formation of bicarbonate with ATP. So you use up energy. And so bicarbonate is prone for nucleophilic attack of your ammonia, forming your carbamate. And in attachment of another ATP, so energy consuming siya, you have your activated form, which is the carbamyl phosphate, and this now can exit your mitochondria. Ito. Ayan. So, what happens here, ayan, ito yung kanina, inside the mitochondria, now you have the glutamate, and this glutamate will will give off your ammonia. So, this ammonia should be converted to carbamyl phosphate, and this carbamyl phosphate, reactive carbamyl phosphate, will react with uh, ornithine, with ornithine to form citrulline. Si citrulline naman is the one na compound that can exit your mitochondria, which will undergo urea cycle. Okay. So isa-isahin natin. So we have explained what happens to your ammonia. So using the carbamyl phosphate synthase, carbamyl phosphate synthase, and have your carbamyl phosphate. Actually, ng carbamyl phosphate natin. Carbamate phosphate. Mali. So, yung carbamyl phosphate. And H2. Sino ba ba? Ito pala. Meron pa. Ito pala. Carbamyl phosphate. Carbamyl phosphate reacts with ornithine. Si ornithine is an example of amino acid, pero hindi natin siya kino-consider for as essential or non-essential. So, since hindi siya need in different processes like yung need na amino acid. Pero it is considered also as amino acid dahil meron siyang nitrogen and carboxyl uh, group. Okay? So, ornithine reacts with um, carbamyl phosphate. So urea cycle para siyang citric acid cycle. So dito is si oxalate. So si ornithine naman dito will react with carbamyl phosphate to form your citrulline. So citrulline now is allowed to exit the mitochondria. Oops, mali. Yeah. Okay? Now, yung citrulline mo should be activated. So, how to do that is you introduce an ATP. So, ATP now, containing, so citrulline AMP intermediate. And upon addition of your aspartate, si aspartate will come from this formation, transamination from oxaloacetate to aspartate. And now you have your arginosuccinate. Okay, so this arginosuccinate give off your carbon skeleton, which is fumarate. Fumarate will undergo to different catabolytic pathway. So now you have your arginine. So si arginine, you have here the two nitrogen containing, and upon addition of water, you can have your urea. So it attacks here to give off your ornithine. So now you have your ornithine again, and this ornithine can react with another carbamyl phosphate inside your mitochondria para mag-proceed ulit yung urea cycle. Okay? So now you have your urea. So this urea is excreted in urine. So that is present in urine in our bloodstream para magpunta sa kidney, and it will be excreted as a urine. Okay? So, I think may mga makikita kayo ditong familiar na mga compounds. Ayan, si fumarate lang naman. Tsaka si, mamaya pa to. Arginosuccinate. Okay? Right, so what happens here? There's the formation of arginosuccinate. Okay, so, ay yung synthesis pathway niya on formation of your arginosuccinate. So, I need to make highlight of it kasi si arganosuccinate, aspartate arganosuccinate is the shunt links between urea and the citric acid cycle. Okay? So, ayan, ito siya. So, within urea cycle and the citric acid cycle, si arginosuccinate can happen from aspartate to arginosuccinate, and yung aspartate can go naman kay oxaloacetate. Okay? So this is the link. So you can link, this is the, parang yung, one of the amazing things that happens sa cells. It can, one of the precursors can undergo multiple catabolic pathway or metabolic pathway given na may mga specific enzymes to do it. Okay? So yun lang yung ina-highlight. Bakit mahalaga si arginine succinate. Alright. So you can see, we can regulate yung urea cycle. So, if ever na, this is the reason din kung bakit minsan matagal yung duration ng excretion of urine, or minsan maikli lang siya. Depende siya dun sa amino acid content ng body mo or amino acid requirement ng body mo. Okay? So carbamoyl phosphate synthase 1 is activated by N-acetyl glutamerate, and upon knowing na depende yung reaction sa glutamate concentration, and it can be also activated by arginine. Okay? So kapag mataas yung glutamate, it can easily form your N-acetylglutamate. Okay? And then next will be the expression of urea cycle enzyme sequences when needed kapag high protein diet nga and in starvation. Depende yung magiging pathway. If ever na mag-excrete ka ng urea, or you will use your amino acid in conversion of your uh, pyruvate to form glucose. Okay? Question so far?
Wala pa po.
Wala. Okay? So what are the genetic defects that can happen in urea cycle? So people with genetic defects in an enzyme involved in urea formation cannot tolerate protein-rich diet. So ang treatment dyan should be um, avoid or ilimit lang yung content, or ilimit yung we call this consumption of the protein-containing diet. So absence of urea cycle enzyme can result to hyperammonemia or buildup of one or more urea cycle intermediates depending on the enzyme that is missing. And ayun. So treatment is careful administration of aromatic acids or in the diet, which can help lower the level of ammonia in the blood. Okay, so these are common diseases in our um, body kapag may missing, pag may genetic defects pala. So this is not common. This is genetic defects in the enzymes related for urea cycle. Okay? So that's yun nga rin yung mga instances whereas we can appreciate somehow yung role ng biochem in order to identify which are the causes of these diseases. And as we can see here, kapag specifically only one of the enzymes ang missing, it can give off a very serious diseases or illness. Okay? Ayan, limitations on amino acids availability. So we have, na not all amino acids can be synthesized in humans. So may mga uh, amino acid that should be obtained in our diet. Okay, consumption of a variety of foods supply all the essential amino acids, including vegetarian only diet. So, you know, these are the non-essential. So we can synthesize these compounds sa ating body, while the essentials are this 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay? Conditionally essential, kasi sabi dito, required to some degree in young, growing animals, and sometimes during illness. Okay? Right. What does it do this? These are the cofactors involved in your amino acid oxidation. One example is, hindi ko ata na-include dito yung isa. Okay. First is tetrahydrofolate or THF. So tetrahydrofolate is formed from folate, an essential vitamin. THF can transfer one carbon in different oxidation states. It's either in methyl, in alcohol, or in aldehyde. Used in a wide variety of metabolic reaction, carbon generally comes from serine and forms interconverted on THF before use. So this is your tetrahydrofolate. Uh, it has 6-methylpterine moiety para amino benzoate. Ayan, in para position sila. And your glutamate. Okay? So what happens here? Okay, you have your tetrahydrofolate and in serine hydroxymethyl transferase, you have your methylene tetrahydrofolate and addition of NADH, you have your CH3. Okay? So depende dun sa need nung amino acid, it can either give off here yung methyl or yung alcohol or your uh, aldehyde. Depende dun sa need nung amino acid. Okay? Or nung process. Next cofactor is adomet or S-adenosylmethionine. Pero we name it as SAM. Yan, S-adenosylmethionine. Ito naman yung better compound for transferring CH3. I mostly encounter SAM in methylation of biosynthetic compounds. S-adenosylmethionine is the preferred cofactor for the methyl transfer in the biological reaction. Methyl is 1,000 times more reactive than CH2. Okay? Kasi ang nangyayari dyan, well, uh, very, dahil sulfur yung, ayun na pala dito. Dahil sulfur, ayan, ito yung CH3 na binigay ko. Ayan. Dahil sulfur yung katabi niya, it is a very electronegative compound. So yung electron pull is papunta doon. So this is a very good electrophile, and once a negatively charged nucleophile attack this CH3, it can easily give off yung bundle. Okay? So synthesized from ATP and methionine, the generation uses N5-methyl THF. Ayan. Yun lang naman siya. Okay? Sisa. This is the ano dito? Formation of your sum. Ayun na, formation of sum. Right. Alright. So, ano naman, after, we have discussed already yung nangyari dun sa amino group, yung after removal of your amino group sa amino acid mo, now you have your carbon skeleton. So, what happens to your carbon skeleton? Depende doon sa form nila. Okay, so this is the summary of your end products. It can be intermediate for the central metabolic pathways. Yan. Intermediates for the central metabolic pathway. Some amino acids results in more than one intermediate. So it can be intermediate for many starting material or precursor for your citric acid cycle, for your for your glycolysis. Ayan. Ketogenic amino acid can be converted to ketone bodies. So ito yung mga compounds that can be converted to acetyl-CoA, mga precursors for citric acid. Example is leucine, isoleucine, threonine, lysine, phenylalanine, tyrosine, and tryptophan. Glucogenic amino acids naman can be converted to glucose as prior for glycolysis. 6 are for pyruvate. So marami siyang entry point. So pyruvate, either pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxaloacetate. Right? Yan. Example of the amino acids that can be converted to pyruvate. Ito, yung kulay-pula. Alanine, cysteine, glycine, serine, threonine, and tryptophan. So as you can see, si tryptophan, nag-exist siya to formation of acetyl-CoA, acetyl-CoA, and pyruvate. And can also, ito, tryptophan. As we have mentioned, na pwede yung amino acid can result in one or more intermediate. Okay? So let's dig in details. Ayan. Degradation of amino acid to acetyl-CoA. So 7 amino acid can be degraded to acetyl-CoA. Acetyl-CoA, right? So what happens here? Mhm. This is not a too detailed information for the conversion of your amino acids to different ito yung precursor since meron dito yung mga shortcut. Meron dito 9 steps which we will not discuss pa. Pero ang gist dito is that yung mga carbons labeled in red, ano ba to, red ba to? In pink, is proceed to formation of glucose uh, neogenic uh, pathway, so formation of your mga acetyl and acetyl-CoA, while yung blue is to formation of mga pyruvate or mga for acetic acid, for the citric acid. Okay? So si tryptophan, involving 9 steps, it can be converted to carbon dioxide and ketoadipate. Itong ketoadipate form niya can be converted to glutaryl-CoA in addition of your um, CoA enzyme and addition of and reduction of your compound. Now, upon release of carbon dioxide in 4 steps manner, now you have your acetoacetyl-CoA which can be converted to acetyl-CoA. Okay? Oops. Okay. Si alanine naman, you can easily convert it to pyruvate by just removal of your amine group. Okay? This is just an introduction of water. Okay, sino pa? Si phenylalanine naman, so it is a ring containing, so meron siyang benzyl group. So it can be converted first to tyrosine. Then yung tyrosine, and OH lang naman yung difference ni phenylalanine at ni tyrosine. And si tyrosine can be converted to fumarate, which will enter your citric acid cycle, and acetoacetate. And upon addition of your CoA, now you have your acetyl-CoA, then mag-form siya ng acetoacetyl-CoA, nag-form ng acetyl-CoA. Okay? Si leucine naman, you just convert it to acetoacetate and directly to acetyl-acetate. While isoleucine, ayan, mga branch compound, by just releasing of carbon dioxide and conversion to propanil-CoA, now you can have another component, yung succinyl-CoA, which can enter your uh, glycolysis. Okay? Si tryptophan, it can either go on dun sa formation of your different starting precursor, pero it can also be a precursor for formation of different biologically important compounds. One of it is nicotinate, or a precursor for the uh, nicotine adenosine diphosphate. Ayan. So, nicotinate. Ito. And it can also go for formation of indoloacetate, a plant growth factor, and serotonin. Ayan. So, familiar kayo sa serotonin, a neurotransmitter. Happy hormone ata ang serotonin. Right? So, ayun. So, yung tryptophan nyo, once na niremove mo yung carbon skeleton niya, yung kanyang indole moiety can furtherly be modified in order to form different metabolic compounds. Or not metabolic compounds, biologically important compounds. Okay? So what are the genetic defects that can happen in our amino acid oxidation? One of it is, eto, naririnig ko to lagi nung nag-masters ko, yung phenylketonuria is caused by a defect in the first step of phenyl degradation. So this is very common disease, and it caused by the buildup of phenylalanine or phenylpyruvate, which causes to impairment of our brain, and that can lead to intellectual deficits. This can be limited by uh, control of your intake of the phenylalanine rich diet. Okay? So, ito yan. So what happens here is that yung phenylalanine mo cannot be converted to tyrosine. So, dahil wala kang enzyme ng phenylalanine hydroxylase. So, what will happen is yun nga, build up of phenylalanine, which is very not good for the body. Okay? Another diseases that can happen is yung tyrosinemia. So, hindi, ano ka naman, marami ka namang tyrosine sa body due to the uh, trisomy 2. Tatlo siya. 1, 2, 3. Dahil sa build-up mo ng mga tyrosine. Okay? At yung isa is alkaptonuria. So alkaptonuria produces few ill effects compared to phenylketonuria, but although large amounts of homogensates are excreted, and its oxidation turns the urine black. So yun na may nangyari sa kanya. It makes the uh, large large content of homogensate will make yung urine black. Okay? So yun yung effects. Yun yung mga genetic defects. Kapag may enzyme missing sa body, it can easily give a harmful or abnormal function sa body. And one of it, yung example nga, ito, phenylketonuria, alkaptonuria, and uh, tyrosine anemia. Depende dun sa magbi-build up ng mga amino acids. Right? Another degradation pathway for the amino acid is the formation of pyruvate. So pyruvate will be the starting material for the citric acid cycle. Ayun. Okay. Formation of citric acid cycle. So tatlo yan. It will can either be hydroxylation to serine to pyruvate. Haven't included that one sa slide ata. Wait lang, class. Questions so far? Wala pa naman. Okay. Wait lang. Hindi ako nailagay.
Sir,
Hmm.
yung po sa urea, production po ng urea. Ano po,
Sorry.
ano po yung yung relationship ng urea sa uric acid po?
Yung formation ng urea and uric acid.
Apo.
Wait lang. Balikan natin yung slide. Ayan. May different pathways wait lang, si amino group, since ang goal nga ng mga nagpo-form ng uric acid is uh, tawag dito, is the conversion of water. So, ang goal nila is to uh, conserve yung water, and they will form, or they will build yung many moles of ammonia, or or urea into one compound. So as you can see here, si uric acid is just a uh, parang cyclic form of four moles of urea. So yun lang siya. And this uric acid is insoluble. Uh, Hindi ko tanda kung wala, hindi ko pala siya mailalagay dito. So, uh, I'll look on the book regarding dun sa conversion ng urea or ng amino acid, ng amino group to uric acid. Pero as you can see here, ayan, so si nitrogen lang naman, ito isang ammonia yan, isang urea, ito isang urea. Okay? And another carbon skeleton just to form your uric acid. So parang naka-form ka ng indole-like compound. Uh, Ayun, yun yung relationship niya. And this is just formed in order to conserve water kasi we know na urea is a very soluble organic compound to water, and yun nga, for conversion of conversion, not conservation of water, pinipili na mag-form ng uric acids. May mga diseases din na can be uric acid are insoluble. So familiar kayo dun sa mga diseases na kapag nasasaktan daw ng buto, ganun, pag pinagsasaktan ng kasu-kasu, hindi ko alam kung familiar sa term ng kasu-kasu, ay sasabihin sa'yo na may mataas yung uric acid. That is actually true kasi uric acid is insoluble to water and it can form salt. At yung uric acid na yon yung salt na yon can stay sa mga uh, buto-buto, and it can cause pain. So, yun din yung cons naman kapag too much yung uric acid na meron sa body. Okay? So, I can siguro give me time to uh, check on it. Limot ko na kung how uric acid is formed. Okay?
Na-presume ko po kasi na si urea po ay salt, parang salt form po ng uric acid.
Ano na?
Si na-presume ko po kasi na urea is parang ano po, salt form ng
Salt
uric
form
acid.
ng uric
Parang
acid.
carbonate tapos carbonic acid, parang ganun.
Right. Yun din siya. Carbonic form si urea. Pero
Okay,
yun.
thank you po.
Okay. Ang difference
Okay na
po
po.
nila ay solubility.
Okay po.
May ilalagay lang ako dito sa lecture slide na. Kita. Ano ba yan? Visualize. Okay, hindi ko siya malalagay. Okay. Ayan. So, degradation of amino acid to pyruvate can be ito yung example niya. For threonine, for glycine, serine, tryptophan, cysteine, and alanine. Okay? So, si threonine can undergo reduction to form your 3-amino 3-ketobutyrate. So, you have here not reduced. This is oxidized form. So, nag-form ka ng carbonyl here, and upon addition of your coenzyme A, Now you can have your acetyl-CoA and this intermediate. So this intermediate can be reacted with your folic acid and then to form your serine. Yung serine naman will undergo different uh, dehydration process to form your pyruvate. Si pyruvate will enter the citric acid cycle. Okay? So, si glycine naman, ayan, may steps uh, hindi ko siya ma-include ata, that will be involved to form this glycine to uh, glycine is just a hydrogen. So, yeah, mag cleave siya to form your carbon dioxide and your ammonia by just simple oxidation process. Okay? Si tryptophan naman will remove its indole and yung carbon, and we have explained din kanina ito. Then yung carbon skeleton niya here will be converted to alanine. Then yung alanine mo will enter your citric acid cycle by converting it to pyruvate using alanine aminotransferase. While si cysteine naman, si sulfur-containing compound mo, will be converted to pyruvate. Okay? So this just includes removal of your amino group and abstraction of your sulfur-containing compound. So this is just addition of water para mawala si NH3 and abstraction of sulfur. Okay? Sir, glycine cleavage. This is just idea on how what happens on formation of pyruvate. Hydroxylation to serine to pyruvate. Ayan. Glycine cleavage enzyme. So apparently major pathway in AMAMA. Separation of three central atoms. Formation of carbon dioxide and ammonia. Ethylene group is transferred to folate. So yung PHF mo will capture yung methyl group from your glycine, and yung methyl group na to will be transferred to your ito, uh, this compound and form your serine. Okay? Lastly is the amino oxidase. So relatively minor pathway ultimately oxidized to oxalo acid. Oxalate. Okay? Another way of degradation of amino acid is the formation of your alpha ketoglutarate. So arginine, histidine, proline, glutamine are the amino acids that can be converted to form your alpha ketoglutarate. So si arginine will just remove your amine group, one of its amine group, si arginine, to form your urea and to form your ornithine. So ornithine will be involved to the aminotransferase from by usage of ketoglutarate to form your glutamate. Now you have your glutamate semialdehyde, and it can be converted to pyrroline 5 carboxylate by just internal formation of pyrroline. So si proline naman, we just remove yung water to form this pyrroline. Tapos balik na siya sa glutamate semialdehyde, which is the uh, starting material for formation of your glutamate by just uh, oxidizing it by NADP. Okay? So si glutamate will move forward to ketoglutarate since marami siyang carbon-containing skeleton. Si glutamine naman, you just remove your amine group to convert it to glutamate. While si histidine has a uh, ring, nitrogen-containing ring. So initially, you just remove your ammonia here, and then many introduction of water to convert it to glutamate. Okay? So, itong carbons na to ang matitira in order for you to have your glutamate. So, nare-remove itong part na ito or histone. Yan. Another is the formation of succinyl-CoA, which is involved to glycolysis. So, si methionine, threonine, isoleucine, and valine, yung four amino acids which is involved sa formation ng succinyl. Okay? So, what happens here? Si methionine is converted to homocysteine. So, what happens there is just remove your alkane methyl group here. Ayan. Then, you form, in addition of serine and removal of cysteine, you have your ketobutyrate. So, this ketobutyrate is the end product naman ni threonine. So, may OH containing group. You just remove your water here and then remove your ammonia. Now, you have this compound which will undergo to different dehydrogenase uh, pathway to form your propionyl-CoA. So, si propionyl-CoA, we have encountered this na sa citric acid cycle. Pero this is a end product of valine and isoleucine. So isoleucine can be converted to acetyl-CoA and yung remaining carbon structure niya can further go converted to succinyl-CoA. So ito yung common ground ni valine. Si valine naman will be oxidized to remove your carbon dioxide twice to have your propionyl-CoA, and with addition of bicarbonate, you have your methyl malonyl-CoA. Then, in presence of your B12, you have your succinyl-CoA. Si succinyl-CoA will enter our glycolytic pathway in order to form your glucose or your pyruvate. Okay? Ayan. Leucine, isoleucine, and valine are oxidized for fuel. So, branch amino acids naman ito. Well, we have encountered kanina sila. And we just have to highlight here na sometimes yung may certain certain disease that that is correlated in the uh, conversion of this branch amino acids, and kapag hindi sila na convert to this specific starting precursors or acyl CoA, uh, acetyl CoA, ayan, they can accumulate and can give abnormalities dun sa blood. So specifically, it is the maple syrup urine disease, accumulation of branch amino acid in the blood and spill over into urine. So nangyayari sa kanya, you have your maple syrup. Kaya siya maple syrup kasi the smell of the urine daw is similar to the odor na maple syrup. Okay? So if it is not untreated, the disease results in abnormal development of the brain, mental retardation, and death in early infancy. Ang treatment is the rigid control of the diet. You have to uh, limit yung pag-intake ng mga branch amino acid to the minimum requirement to permit normal growth. So yun yung mga challenging part on how will you, I mean, di ba? You can minimize your diet if you have enough resources to do it. Kasi uh, you can do it. Parang you can monitor yung ano ba yung kailangan mo lang i-intake and i-consume. So, medyo challenging siya, especially dun sa mga taong hindi naman nila kayang i-monitor yung yung mga amino acid intake nila. So, parang walang means to do it. So, ayan, yun yung mga disadvantages, mga pros and cons. Magkaroon ka ng mga kasi may mga enzymes naman din. I mean, pag may encounter natin siguro sila sa there are some enzyme lacking uh, mga illness na hindi naman siya, hindi grabe yung mga uh, effects. Like you just have a missing, dito, yung sa mga albinism, like ang effect nun ay uh, nagiging white or you have lack on melatonin. So, ang nangyayari, kulang ng darkness yung skin. So, parang hindi siya ganun uh, kagrabe. Pero some of the diseases like this, yung missing enzyme in conversion of certain amino acid, it can really affect yung normal way of living, especially yung normal growth, yung growth and uh, thinking or knowledge or mental health or not mental health, mental capacity no may body. Okay? Yan. I think this is the last example of degradation of amino acid to oxalic acid is to formation of oxalic acetate. So dalawa lang sila, si asparagine and aspartate. So this is just ano na naman. Si aspartate, ang pinagkaiba lang naman nila is may nitrogen, another nitrogen group or amino group, si asparagine. So you just remove your amine group, one of its amine group, then si aspartate can be converted to oxaloacetate by just transamination of alpha ketoglutarate and glutamate. Okay? So yun lang yan. These are the, ayan. So those are the degradation pattern or degradation pathway for the conversion of your amino acids to different precursors for citric acid cycle or glycolysis. So some of these compounds can form multiple intermediate or can form specific intermediate na lang given to their carbon skeleton. Okay? So some of these compounds involve multiple steps or just a straightforward conversion of their structure. Okay? So some of this just release carbon dioxide and converted them to ammonia and then release it as a form of urea or uric acid or ammonia only. Ayan. So these are the summary of this amino acid oxidation. Bukod dun sa amino group that we have discussed earlier that can be formed to urea and uric acid or ammonia, yung carbon skeleton na remaining ng amino acid can be furtherly converted to small precursors of different catabolic pathways, specifically for citric acid and glycolysis. Okay? So the last slide includes only the uh, some human genetic disorder related to amino acid catabolism. So ayan, albinism. So hindi rare siya somehow per 3 per 100,000 births. Ang defective process niya is the melanin synthesis from tyrosine. So you have defective enzyme which is tyrosine 3 monoxygenase, and the symptoms and effects is lack of pigmentation. So you have white hair and pink skin. We have discussed alkaptonuria. Ayan. Another example are argininemia, argininosuccinic acidemia, and carbamol phosphate synthase. So, yan. As you can see here, meron dyan na may mga, eto, can cause early death if ever na meron kang hindi eto, specifically kay carbamol phosphate kasi hindi, especially sa uh, kapag hindi mo kaya ma-convert yung amino group mo to urea, and since ang ammonia is a toxic compound sa blood stream natin, mag-i-stay lang siya doon and it can cause early death. So bata pa lang, it can, baby pa lang, possibly infant pa lang, it can die if ever na there's no carbamol phosphate synthetase. So, rare siya, 0.5 per 100,000, pero that's marami pa rin. For 100,000 to eh. So less than 0.5. So that's around 50. No, not 50. 0.5%. 1, 2, 1. Parang mga nasa 500 in every 100,000. So that's alarming pa rin. Homocystinuria. So, in bone naman siya. Early death din sa maple syrup. Tsaka sa methyl malonyl CoA mutase. Conversion of propionyl CoA to succinyl CoA. Ayan. So these are the examples of genetic disorders lacking or there is wrong uh, conformation in your enzymes, which are important in the conversion of different amino acids. So, ayan. So, these are just the, ito na yung medical approach ng biochem or ng chemistry general on how you will try to solve this kind of diseases. So, one of the treatment kanina is just in control of diet or pwede kang magkaroon ng intake of specific uh, uh, drugs to help na ma-retain yung amino acid na present sa mind. Questions? I think that ends our discussion for amino acid oxidation. May question kayo? Okay. Um, Sige. May question? Okay. Um, ang midterms natin ay this week na, no? So hanggang dito lang sa amino acid oxidation. Tapos, may schedule na ba kayo ng ano yun, ng midterms nyo? I mean, ng ibang subjects? Ilan pa? Usually, ano siya, weekends, weekdays. Weekdays yung schedule niya siguro, I think. Okay. Alright. Sige. I will just post the midterms exam, pero I'll ask muna siguro si Ma'am Joy kung ano yung schedule na pwedeng maalat sa akin. Pero yon hanggang dito lang yung topic. Hanggang dito yung coverage ng topic. And I'll finalize kung ano na magiging type ng exam mo. Pero usually, sa biochem, ang exam ko ay multiple choice. Tapos, you'll answer it in specific time. I will send off yung uh, exam, then you will answer it in certain period, then submit it sa akin agad. So, yun. Multiple choice, tapos may some enumeration or may problem solving. Okay? Yan lang. Questions? May questions pa kayo? Clarification?